Welcome students to the last lecture on chapter 23 on the digestive system. This is going to look at part 3 of this chapter, essentially how digestion and absorption happens in the GI tract. So of course when we were talking about digestion, we were talking about breaking down our foods essentially into these building blocks, because really only the building blocks that our body is capable of absorbing. This is a catabolic process, so a process that breaks bonds. It's essentially breaking the bonds in our foodstuffs and releases energy. It is a hydrolysis process. So we use water essentially to split our foodstuffs. So here you can see our foodstuffs. We add a little bit of water and we break it into those building blocks. This does require enzymes, otherwise this process would happen far too slowly. Essentially just not at a rate fast enough to sustain life. Catalysts are enzymes. They help lower the activation energy sort of without being consumed themselves. So we're able to get from reactant to product much more quickly, and the enzyme can essentially do it over and over and over again. So this is what that hydrolysis process looks like. So here we happen to see 2 amino acids. So we have a dipeptide. You can see the enzyme has a little active site for that particular substrate. They are all substrate specific. So a protein digesting enzyme would not be able to digest fats, for instance. But the substrate binds on to the enzyme. The enzyme sort of changes shape a little bit, makes it just the perfect place to add in some water, which then breaks the bonds between those amino acids. That releases some energy whenever we break bonds. The products are then released from the enzyme, and the enzyme you can see is back to its original shape, sort of ready to do it all over again. So all of our food digesting enzymes are considered hydrolases, what we call hydrolytic enzymes, enzymes that use water to split larger substances into smaller substances. Essentially what we're doing is we're breaking long chains of things into smaller chains, and then we're breaking those smaller chains into ultimately sort of single building blocks. So we're going to go from a polymer to something called an oligomer or a trimer or a dimer, multi, 3 or 2. The enzymes that do that are going to be typically the salivary enzymes, the gastric enzymes, or the pancreatic enzymes. And then we use the brush border enzymes to then break those sort of smaller chains or trimers and dimers into the individual sort of monomer building blocks for our carbs, proteins, nucleic acids, and fats. Absorption is then the process by which we're going to take those now single sort of building blocks and move them out of the lumen of the gut actually into the blood. So all of the cells of the small intestine have very tight junctions between them, so things don't slip in between very easily. So that means pretty much everything that gets absorbed has to go across the epithelial cells. And most of the absorption takes place in the duodenum and the jejunum. It's really mostly sort of finished before we get to that last portion of the small intestine. So the carbs, the proteins, and the nucleic acids follow one mechanism. The lipids are a little bit different. So carbs, proteins, and nucleic acids, they are actively transported into our capillaries of our intestines. And then once they are absorbed, they get shunted to the liver via the hepatic portal vein so that the liver can process them. The lipids, since most of our, of course, intestinal cells are phospholipids, they can be absorbed via passive transport, and they get absorbed into something called a lacteal. And they actually don't get returned to the blood until your lymph fluid returns to the blood. So the lipids do not go through the liver. So carbs, proteins, nucleic acids, active transport to the liver, lipids, passive transport into the lacteal, and they bypass the liver. So if you look at carbohydrates, you can see carbohydrate digestion starts in the mouth. A little salivary amylase. It's going to break down our starches into an oligosaccharide or a disaccharide. The pancreatic amylase then breaks the oligosaccharides and the disaccharides into our sort of lactose and maltose and sucrose. And then we use brush border enzymes to actually get to the single monosaccharides. And then it's those monosaccharides that are absorbed via active transport. So you can see here's how's that's happening. So here we've got a little bit of starch, some disaccharides, here's some pancreatic amylase, helping break down that starch into some disaccharides, and then we're using these brush border enzymes ultimately to get our 2 monomers, and then the monomers are active transport. So the way this active transport works is we use the sodium-potassium pump down here on what we call the basolateral surface, the surface closer to the capillary. That's going to pump sodium out of the intestinal cell constantly. That creates a lower concentration of sodium inside the cell than in the lumen of the intestine. So sodium then uses a transport protein to diffuse down its concentration gradient, and that brings the monosaccharides with it. We then use another carrier protein to absorb those monosaccharides ultimately into the capillary. Protein digestion starts in the stomach with the pepsin, requiring, of course, the presence of hydrochloric acid. The hydrochloric acid is going to unfold the proteins, denature them, allow the pepsin to take those polypeptides and break them into slightly smaller chains. And then we're going to use some pancreatic enzymes like trypsin and chymotrypsin from the pancreas to break down those smaller chains into even sort of smaller pieces in the small intestine. And then ultimately the brush border enzymes are going to get us to our single units. So here you can see, here's your little amino acid fragments. They've already been broken down into sort of fragments by the pepsin in the stomach. The pancreatic proteases break them down even further, and then the brush border enzymes get us down to single amino acids. Again, we use the sodium-potassium pump to actively pump sodium out of the intestinal cell into the sort of interstitial fluid of the intestine. That creates a low concentration of sodium inside the cell, then in the lumen. So the sodium diffuses down its concentration gradient, and the amino acids come with it. And we use another carrier protein for those amino acids to get out of the intestinal cell and ultimately into the capillary. And then nucleic acid digestion is pretty much entirely confined to the small intestine. We're going to use pancreatic enzymes to break the nucleic down acids into their single nucleotides, and then the brush border enzymes to break the nucleotides into the actual pieces of the nucleotide. So the base, the phosphate, and the sugar. So here you can see, here's our nucleic acid, some little RNA for us. The pancreatic nucleases break us down into our nucleotides, and then the brush border enzymes break us down into our pieces of nucleotides. Again, active transport using that sodium-potassium pump and the carrier proteins into the blood, off to the liver. So very similar process between the carbs, the proteins, and the nucleic acids. Sort of the weird one is the lipids. There is a little bit of lipid digestion that occurs in the mouth with sublingual lipase. There's a little bit of lipid digestion that occurs in the stomach with some gastric lipase, but most of the digestion of lipids takes place in the small intestine with bile from the liver as well as the pancreatic lipases. So again, we have to emulsify the fat first with the bile. We need to break that big fat droplet into much smaller fat droplets. That allows more surface area of droplets in order for the enzymes to work. The pancreatic lipase will break down these fat droplets into sort of triglycerides and monoglycerides and little fatty acid chains. But since most of our fluid inside our intestine here is sort of water, they sort of huddle together into something called a micelle in order to travel to the surface of the intestinal cells, where they sort of diffuse passively across the intestinal cells. They are then packaged with some proteins into something called a chylomicron, and it is ultimately that chylomicron that is absorbed into the lacteal, and the lacteal will dump into the lymph system. And so fats, when they are absorbed ultimately into the lymph system, and they do not go to the liver like the carbs, the proteins, and the nucleic acids. So slightly different process for the fats than the carbs, the proteins, and the nucleic acids. Vitamin absorption occurs mostly in the intestines. So in the small intestines, all of the fat-soluble vitamins will get absorbed when any of the fats get absorbed with the micelles, and then the water-soluble vitamins typically absorbed via diffusion as well. Vitamin B12, that very, very large molecule that's necessary for our red blood cells to be made, can only be absorbed by our small intestine with that intrinsic factor. So again, that's that sort of thing from your stomach that only your stomach does. And then primarily in terms of vitamin absorption in the large intestine, it's really mostly just whatever B and K vitamins that your bacterial flora happen to make. Electrolyte absorption occurs all along the length of the small intestine. We can change electrolyte absorption, particularly for iron and calcium, in the duodenum with some hormonal action, sort of based on what the body needs. In terms of water, there really isn't a lot of water that gets excreted with your feces, even though your GI tract makes a lot of water. About 9 liters of water, including sort of all of the secretions all along the GI tract, plus the water from the food and drink. Almost all of that will be absorbed via osmosis in the small intestine, and almost all the rest of that will be absorbed by the large intestine, usually just coupled with some solute uptake. When the smaller large intestine absorbs sodium, for instance, the water pretty much comes with it. So you lose very, very little water via defecation. Most of the water that processes through your GI tract is absorbed ultimately back into the body. And that's it. That is the end of chapter 23.